Hi, my name is Kareena Villalobos, and this is my 918 midterm presentation. Today, I will be discussing the studies by Thomas Jessel and colleagues to show that sonic hedgehog is a secreted morphogen that provides a ventralizing signal. We begin our journey with the notochord of early embryonic development. Scientists had just discovered the notochord was necessary and sufficient for the neural plate to ventralize or develop ventral fates. The next step was to search for what the notochord contained that could be a ventralizing signal. More specifically, scientists were searching for a secreted factor that would be surely expressed in the notochord. Once they found that factor, they then would need to show that the secreted factor is sufficient, or that it is able to send a ventralizing signal. Finally, the scientists would need to show that the secreted factor was necessary for ventralization to occur. Meanwhile, scientists Eric Weishaupt and Christiane Nusslein-Wohlhard discovered that some Drosophila fly mutants developed abnormal anterior-posterior body patterning. Developmental scientists were now determined to investigate the secreted factor behind this abnormality. Thus, sonic hedgehog research surged soon after. Now we know that sonic hedgehog is a secreted morphogen, we can investigate whether or not it was expressed at the right place and time. Soon after Weishaupt and Nusslein-Wohlhard, Cliff Tabin had discovered the sonic hedgehog morphogen in the zone of polarizing activity, or ZPA. Found to be involved in the anterior-posterior patterning of the vertebrate embryo, studies also showed that the floor plate of the neural tube could substitute for the ZPA in the limb of chicks. This discovery led Thomas Jessel and colleagues to investigate sonic hedgehog in the floor plate and notochord. Methods they used included in situ hybridization histochemistry. Using a digoxygenin labeled antisense RNA probe, the single strand was hybridized. Then, after mixing the probe in with the neural tissue samples, the RNA binds in place of the expressed sonic hedgehog. After washing away the unbound probe, they found the sonic hedgehog gene expression as shown in blue. In Figure A, we see restriction of sonic hedgehog expression to the notochord at early stages of caudal neural tube development. And in Figure B, we see the expression of sonic hedgehog by both notochord and floor plate at late stages of neural tube development. Now we know that sonic hedgehog is at least expressed, but is it sufficient? Rowlink and colleagues decided to test this question by extracting parts of the neural plate without the notochord and adding purified E. coli-derived sonic hedgehog. They then left 10 to 12 samples for 24 and 44 hours. After observing the 24-hour samples, the scientists found that the more concentration of sonic hedgehog there was, the more cells were differentiated. Because of the influence of the NT3 protein, though, they deduced that other factors help increase the number of motor neurons. However, when observing the 44-hour neural plates, they found that sonic hedgehog increased the number of cells by 1.4 and 10-fold. Therefore, sonic hedgehog is sufficient to independently induce motor neurons and floor plate cells at different concentration levels. Now the only thing left in our journey is to determine if sonic hedgehog is required for ventralization. Erickson and colleagues decided to use polyclonal and monoclonal, or H4 and 5E1 antibodies respectively, in vitro to specifically block sonic hedgehog signaling, since neither Indian or desert hedgehog are expressed in the notochord. They then isolated neural plate, neural tube, and notochord tissue from stage 10 to 17 embryos and grown in vitro for 48 hours. As seen in the figure below, the authors found that floor plate differentiation and motor neuron generation was blocked in the presence of the antibodies, which indicates that sonic hedgehog is indeed required for ventralization. The scientists also discovered that there are two critical periods for neuron specification, and the concentration determines whether or not they become motor neurons or interneurons. In the early period, naive neural plate cells are converted into ventralized differentiation progenitors, and in the late period, sonic hedgehog drives the differentiation of ventralized progenitors into motor neurons. Now we have completed our journey towards identifying the sonic hedgehog morphogen as a ventralizing signal. Thanks for watching.